All right, thanks everybody for joining. I'm excited to have David Emms from InstaDeep here. David is an AI-focused computational geneticist at InstaDeep. He has a background in math and computer science and carried out 8 years of a postdoctoral research at Department of Plant Sciences at the University of Oxford in Professor Stephen Kelly's lab. There he worked on the C4 Rice project, developed OrthoFinder for large-scale ortholog prediction. That's a tool I've used a lot. So thank you, David. And work with shoot.bio website as a gene tree based alternative to BLAST. Since then, he has been working in applying AI methods to agriculture and drug development and continuing the work on InstaDeep's Agro Nucleotide Transformer models. And I'm excited to hear what David has to share.
Thanks, Carson. So thank you very much for the invite. It's great to get to talk to you all today. So thank you very much for that, and also thank you for all the work you've done on your various genomic resources, which I think I've made use of quite a lot, both in my previous research and in my current job. I guess I'd like to talk about our nucleotide transformer model, specifically about our Agro Nucleotide Transformer model that we've been developing at InstaDeep, and just give a bit of background on that and talk about some of its capabilities and the work that we've been doing. And I guess a lot of this work I'll be presenting is thanks to people like Javier, who developed the AgroNT model, to Hugo and Bernardo, also both at InstaDeep, working on the nucleotide transformer and the segment nucleotide transformer models. Thanks to them for the work here. Fundamentally what we're interested in doing is developing what we call foundational models, which are large-scale AI models, which can perform. Sorry, if you could just give me 1 second while I just check the slides. Yep. Let me share that. Yep. So is that currently being shared at the moment? Are you seeing slides at the moment?
We are seeing just the slides now.
Perfect. Yep.
Sounds so familiar.
They're coming through okay. Yeah. Okay. Sorry about that. I was just confused by an animation. So we're generally interested in building these large-scale AI models that, with a small amount of adaptation, can be trained to take various genomic sequences and perform a wide variety of tasks, be that things like gene expression prediction, chromatin accessibility, and classification of variant effects, and so in a way that sort of makes use of the knowledge between these different tasks, and so really we're interested in building in this case neural networks, but in AI and ML there's many other methods which are basically trained on large amounts of sort of paired data, where we have an input and its expected output. And in this case, I've shown where we have an input as an image of a digit, and the output that we're trying to predict is what digit that is. And we feed this input through a neural network, which is really just a very large mathematical function taking the pixel values at each point in this image and carrying out a series of transformations until you get probabilities at the end for different character digits that this could represent. And so the idea is that we can train one of these models by giving it a large number of such examples, and then we calculate what's called a loss function, which allows us to determine in which direction we need to change the parameters in our model to get it to give us more and more accurate predictions, to more accurately represent the training data that we're presenting it with. And so in the classic machine learning problem of, is this an image of a chihuahua or a blueberry muffin? We're looking at sort of building a model which will take these images, perform some kind of transformation, and return to us a probability of, is it a chihuahua? Is it a muffin? And to do this, what it needs to do is really identify sort of important attributes of the image, in this case, which are going to help it determine the task at hand. Or when we turn to the problem of DNA, we'll need to train the model so that it understands important aspects about the grammar of DNA in order to make predictions on DNA sequences. But the problem is that we often have quite limited amounts of data, and this is getting into the reason for this approach we've taken with these large foundational models rather than more standard approaches. So if we can consider the task of predicting gene expression, given the promoter sequence, you can imagine we've carried out an RNA-seq experiment. We have perhaps the gene expression levels for 30,000 genes in a particular tissue type. And for each of these genes, we have associated the expression level. So we can feed these into a neural network. We can tune our parameters in our neural network to make predictions. And then we have a neural network which can be fed a novel promoter sequence and will predict the gene expression levels for that promoter sequence. But you know, these 30,000 promoter sequences that we've trained it with are sort of quite a small quantity. If you think it would take just 10 nucleotides, a string of just 10 nucleotides to give us 1 million different possible combinations. So if we wanted to be able to predict, if we made a construct with some synthetic promoter sequence and we wanted to be able to predict its expression levels, the model is really going to have to carry out a large amount of generalization to, like I say, to understand the grammar of the DNA in a sense, to be able to make accurate predictions for novel sequences that it's not seen before.
Sorry to interrupt you. I think I don't know if you are still explaining the first slides, but we are like, just see the first one.
You're just on the first one. I've got a slide up saying gene expression prediction task.
No,
is that?
We are still on the first one. Yeah, sorry about that.
Okay, that's not changing when I'm doing that. No, let me just stop there and try that again. Fine. About that. And do you see the title slide currently?
Yes. Try to change it now.
And is that advancing?
Yes, this is okay. Now there is a change. Yes. Oh,
yeah,
okay.
Okay.
Thank you.
We may have missed some things, but is there anything I should clarify at this point, or shall I carry on?
Okay.
I'm very happy, I should say, to take questions throughout the talk. So if anyone does have any questions, then please.
Okay. Yeah, so I think you can go on, and if there are any questions, yeah. we will interrupt you. Thank you, David.
So I hope that wasn't too confusing as I thought about some of the earlier parts then. So yeah, so just to recap, the problem that we're trying to deal with is that we want to be able to take a DNA sequence and something we want to predict about the DNA sequence, for example, gene expression level. But we want the same model to be able to make other kinds of predictions with a few changes, for example, chromatin accessibility. And in order to get around the problem of small amounts of data, in order to build up this kind of representation of DNA, instead of carrying out these kind of training that I described so far called supervised training, we can actually carry out what we call sort of self-supervised training. And this has been very successful in the modeling of human languages in large language models and things like ChatGPT, which we've seen recently, and has also proved very useful in our nucleotide transformer model that we developed. And the idea here is that instead of feeding the model pairs of sequences together with the label that we want it to predict for that sequence, we can just feed it the raw genomic DNA sequences of species that we're interested in. And the way that we carry out this self-supervised training is we mask out portions of the DNA sequence, and we then ask the model to try and predict what it thinks that masked piece of sequence should be. And we do this many times across entire genomes of multiple species. And so what we find is that the complexity, the amount that this task confuses the model over time decreases. It gets better and better at predicting what these masked bits of sequences are. And the only way it can really do that is by building up this internal representation of kind of the grammar of DNA sequences. And so what we can do is carry out this pre-training that I just described with masked language modeling of DNA. And then we can use that model that we've developed to then carry out the prediction of a particular task of interest, such as gene expression prediction or chromatin accessibility. So we take the model that we've trained with its weights associated from that pre-training step. We and put a small, what we call a prediction head on top of that model. And then we find that very rapidly, with small amounts of data, we can train this model to give very accurate predictions, basically piggybacking off the understanding of the grammar of DNA that it's built up to this point. And we can actually see that it really is building up in this kind of self-supervised training, an internal representation of sort of different genomic elements. So here what I'm showing is the pre-training stage, where we just give the model bits of DNA sequence. We don't tell them what this DNA corresponds to. And this plot shows an embedding of different elements according to whether they're UTRs, coding DNA sequences, intergenic. And we can see that as the training progresses, the model actually separates these different elements out from one another, despite, as I say, not being told in advance what they are. So it's building some kind of sophisticated internal representation of the DNA. So yeah, this is the process that we use to develop our AgroNT nucleotide transformer model. It was pre-trained on 48 plant species, selected either for being model plant species or important crops. with I think about 1 billion with 1 billion parameters, and training it on 380 gigabases of nucleotide sequences. And so this is just a summary of that, the input data, this pre-training that we do, and then the fine-tuning of the model for specific prediction tasks. So to look at one of these tasks that we trained it on, we were looking at promoter strength prediction. So we used some data from a StarSeq analysis from maize, sorghum, and Arabidopsis, looking at transient expression in maize protoplasts and in tobacco leaves. So we had sequences attached to minimal promoters, and the task was to predict the expression levels that would be observed for each of these sequences. So this is based off 160 base pairs of sequences extracted with reference to the transcription start site. So 165 base pairs upstream of the transcription start site and 5 base pairs downstream of the transcription start site. And we see that this pre-trained nucleotide transformer model outperforms the benchmark CNN from this paper for each of the species across the two models. So in preparing the AgroNT paper, we prepared basically a plant genomics benchmark of various prediction tasks, such as alternative polyadenylation sites, splicing sites, gene expression, identification of long-coding RNA, and in almost all of the benchmarks, the AgroNT model outperformed the benchmark method or matched it in accuracy. So I think next I'll just talk a bit more about the work on the gene expression prediction task. But at this point, does anyone have any questions that they'd like to ask before I go on to that? Or if we're all okay, I shall continue in that case.
David, there is one question, I think. Tanner, if you...
Sorry, you mentioned that you're doing edible genomes, but there's Arabidopsis and tobacco there.
Yeah.
I wonder if if you included wheat and barley.
Yeah, wheat and barley were both in there. So the Arabidopsis and tobacco were selected as often model species, important to genomic research in plants.
Oh, okay, makes sense. Thanks.
And yeah, we're now working on an updated version which has a larger range of crops as well. So yeah, so in the gene expression task, I think it's an interesting task because there's a lot of data, and it's quite an important for research, and also in agronomics more generally. So if we can predict accurately the expression levels for given promoter sequences, this gives us a way of carrying out rational design of promoter sequences for crop improvement programs. So the data set we compiled was across multiple species and multiple tissue types in those species. I think from about 7 tissue types in rice to, I think it might have been as many as 56 tissue types in Arabidopsis. And so on the right is a sort of a representation of the kind of data we're looking at, where most in a particular tissue, a large proportion of the genes have 0 expression or very low expression, and then we have a second high expression mode of the genes which are expressed, and in some cases this second mode is nicely distinguished from the low expression mode. In some of the other tissue types, these merge into one another slightly more. So for this data set, we applied a consistent data preparation to quantify read counts across the different tissues and different experiments that would be used for the task. And for AgroNT, we have a context window of 6 kilobases. So AgroNT will take as an input a sequence of 6,000 nucleotides. So we designed a data set based on 5,000 bases upstream of the transcription start site and 1,000 bases downstream of the transcription start site. The other task to be, or point to be aware of, is this controlling for sort of data leakage across our training data set compared to our test data set. So you can imagine if we randomly assigned genes to either training, validation, or test, then you may have one gene in training, which is closely related, it's a power log of another gene in the validation data set. So we followed standard practice of clustering these genes into the gene families, using both using a blast all versus all search, constructing a graph of homologous genes, and then clustering the genes based on that graph in order to cluster the genes into their gene families, and then split our data such that all the genes from a particular gene family always fell entirely within one of the classes, be that training, validation, or test, so that we didn't end up with 2 very similar sequences, one in the test set and one which was used for the training. And so we have shown the graphs here of the observed gene expression versus the predicted gene expression for a number of tissue types here, showing both a heat map and a contour plot. And we see that we're achieving quite high R-squared values for agreement between the predicted and observed values, ranging from about 0.57 to 0.68, and we get good accuracy across a large range of tissue types. So to benchmark this against other methods, we looked specifically at the task of not predicting the absolute expression value, but instead determining whether a gene was expressed versus unexpressed, which matched the task in the Washburn and et al., I think 2019 paper, from which we'd obtained the gene expression data, and looking at the area under the receiver operator curve, we found that the AgroNT model outperformed the Washburn CNN model predicting whether a gene was expressed or unexpressed, both based on the either promoter sequence or the terminator sequence. So then we dug in a bit more into what controls the accuracy of our prediction methods of our predictions. So we had a couple of different ways of predicting gene expression. One was as a categorization setup where we took the observed expression and we put those genes into bins. I think around 50 to 100 bins of the most lowly expressed genes up to the most highly expressed genes. And then the expression prediction task becomes one of categorization in terms of placing a gene in its correct bin according to how highly expressed it is. But another approach we took was to look at a regression model where we predict directly an expression level for a gene. And it's this regression problem where we're predicting directly the expression level that I'm looking at here. And in order to construct this, we developed a 3-state model where we have what we could call the hurdle, which determines whether a gene is completely unexpressed or not. And then for those ones which are expressed, we determine, is it in a low expression state, the grey plot here, or a high expression state. And then finally, once we've assigned it to one of those two distributions, we then have our regression model, which predicts an actual expression value from the sequence based on whether it's the lowly expressed or highly expressed gene. So at this stage, we had approximately equal accuracy from both using this expression model here or the categorization model that I described before. But the advantage of this regression model is that it gave us a way to sort of examine where different contributions to the accuracy of the model occurred. So we could look at does the drops in accuracy from perfect accuracy come from whether we decide whether a gene is unexpressed or expressed, or does it come from the prediction of the numerical level of expression given these? And we could see that very few of the errors come from deciding whether a gene is completely unexpressed or is expressed. Our model was able to quite accurately predict that, but more of the errors come from this second state prediction of are we in a high expressed or low expressed state, to the extent that we saw that if we could predict that completely accurately, that would boost our overall performance from what we were seeing in this particular case of an R-squared value of 0.52 to a potential upper limit of 0.75. So indicating that a lot could be achieved if we can design the model so it's better able to distinguish between genes which are in this low expressed state versus in the high expressed state. Another avenue that this gene expression prediction model opens up to us is to look at the sort of positional importance of sequences across the promoter sequence. So to do this, we can take a particular promoter sequence. We can look at a particular region in it, and for example 6 nucleotides, and we can shuffle those 6 nucleotides, and we can predict the gene expression before we carry out this shuffling task and afterwards, and we can look at how large the change in expression is as a result of this change in the sequence, and then we can therefore determine if the sequence at a particular location is high or low importance to the expression levels that we observe. And so carrying this out across the whole length of the promoter sequence, as we see, we sort of recapitulate what we generally expect, but with quite high precision or ability to look at this in a site-specific way, seeing that that most of the contribution comes from this region very close to the transcription start site, and in fact most of this, the largest effects are just downstream of the transcription start site in our analysis. And likewise, we can look at the different prediction accuracies obtained from taking different 6,000 base pair windows with respect to the position of the transcription start site, from this 5 kb upstream one to this 1 kb upstream, 5 kb downstream of the transcription start site. And we find that actually taking the majority of our sequence from the gene itself, rather than upstream of the transcription start site, actually boosts our accuracy quite significantly across all tissue types. So yeah, I guess again I can take any questions there, or and after this I'll go on to talk about our newer segmentation model. So in that case, I'll just carry on, and so everything I've presented so far revolves around models which take an entire DNA sequence and produce a single prediction from that that sequence. But what we're also interested in is predictions at a nucleotide level. So if we pass a sequence in, we can then make predictions at each individual nucleotide. And this is particularly important, for example, genome annotation, to say which nucleotides correspond to an intron versus which regions correspond to an exon. So in developing the our segment-NT model, we actually started by first making some architectural improvements to our underlying nucleotide transformer. So as I described previously, our AgroNT model has a context window of 6,000 base pairs. The version 2 of our nucleotide transformer used a more efficient architecture, and it allowed us to increase the context length to 12,000 base pairs. And also this more efficient architecture actually allowed us to obtain similar performance with a model of only 500 million parameters to that obtained with a 2.5 billion parameter model on the previous architecture. These data I'm presenting here are from our human and multi-species nucleotide transformer model, rather than the AgroNT model I've mainly concentrated on up to this point. But they illustrate the point about the architectural improvements for the parameter efficient model underlying segment-NT. And yeah, so the idea of segment-NT is it's this model which will provide nucleotide resolution prediction, allowing us to perform tasks such as genome annotation. And so the idea is that we can pass a sequence in, and I think this is a human sequence, And what you can see is we get out predictions of the probability at each nucleotide, whether it corresponds to a protein coding gene, a UTR, exons, introns, etc., splice, donor, acceptor, and sites, etc. And so, yeah, and we're currently working on a plant-specific version of this to perform genome annotations in plants as well. And just to sort of talk about a few of the architectural changes as well, we also tested this longer sequence length still, so up to 30 training model up to 30 kilobases, and we saw that in all of these tasks of annotating the genome, the accuracy of the model increases continuously as we up to 1,000 base pairs as we increase the amount of the DNA sequence that it sees as context when making predictions at the nucleotide level. And we also looked at sort of rescaling, which gives us the ability to train on sequences of 30,000 base pairs, where this training is the more costly task in terms of computation and memory requirements, and then rescale it to actually take in longer sequences still. And we observed that we're actually able to extend the sequences up to 50,000 kilobases, achieving higher accuracy still in our prediction tasks. So this is just to briefly show the splicing prediction of the model, where we were able to accurately predict splice, donor, and acceptor sites, and to do that with higher accuracy than Splice AI, both on its test set and on a test set we prepared, which uses more realistic sort of whole genomic sequences from which to make the predictions. So this model, we have currently both a human model and a multi-species model for segment NT, but we also examined, well, how well does this actually generalize across more and more distant species. So we see with the human model, we attain, for most tasks, quite high accuracy, while remaining in, for example, the tetrapods or within the animals, with slowly decreasing accuracy. And actually, by the time we get to plants from the human chain model, the accuracy is starting to drop off significantly, which is the motivation for wanting to develop a plant-specific version of this model. But it does show the generalizing ability of a model trained with one species to make predictions in other species which it's not seen. So yeah, that was, I guess, a whistle to stop of our AgroNT large language model for prediction on various genomic tasks, pre-trained on plant genomic sequences. And yeah, I talked a bit about the fine-tuning to perform these different tasks after carrying out the pre-training on raw genomic sequences. And this gives us, leading accuracy in tasks such as gene expression prediction. And yeah, we're extending this to look at training on specific regions, on large amounts of genomic data, on larger context windows, so that the model can take into account more of the genomic context around the region of interest we want to make predictions, and with more efficient architectures to give faster models. And so you can try out these pre-trained models. For example, I think Hugging Face is probably the easiest way to sort of experiment with the models, for example, generating embeddings from AgroNT. And you can also find the plant genomics benchmark set associated with our paper. But yeah, I think I'll stop there and happy to answer any questions.
Thank
Yeah.
you, David. A little, yes.
Hey.
Hi, thank you. This is really fascinating. So I'm really kind of interested because we've just been working on a re-annotation of the Arabidopsis genome and using actually gene predictions that were generated from MCBI. And they definitely seem to have some trouble with some plant-specific gene families. So I'm kind of curious as to, why you think that your well, one is, there were two questions. One is about like sort of, why it sort of, fails as you get farther away from the human genome. And then also I'm curious about what your sort of input sequences are. Like, for example, for Arabidopsis, what's your reference genome? Two questions.
Yes. So I guess the degradation in performance is, I think, largely down to the fact that it's seen the genome of one species, and the further away we get phylogenetically from that species, the less it can correctly generalize between how what fundamentally what the genes look like in one species and another species, because the genetic machinery changes, codon preferences change, things like this. And so training it with genomes directly related or closely related to those species we're interested in making predictions for, I think is fundamental to achieving the highest accuracy with this kind of approach. And you remind me the second question?
Oh, I was just actually wondering what the inputs were for, like what did you use for an Arabidopsis reference?
So we're using, so in that work I presented there wasn't actually any training on the Arabidopsis genome, but we are looking at that now. So I think we're using our report 11 annotation of the Arabidopsis genome, and we will, we're looking at training on probably in excess, somewhere between say 100 to a few hundred genome annotations, looking at the taking into account the quality of the annotations that we have available. And I think probably the argument I would give for this, giving better annotations as predictions, is that is the ability of the model to generalize between the gene models it sees in one species and the gene models it sees in another closely related species. And obviously a lot of annotation methods these days do that, but I think in some ways it's quite hard to formalize all of these kind of insights of, well, this is what a gene looks like in one species, and this is what a gene looks like in another species, and this is how you can generalize across them. I think we're hoping that rather than it being a sort of a more expert curated method of sort of trying to express this problem definition, we rely on the kind of the training loss of the model and the ability to try fine-tune this model to give more accurate annotations.
Thanks.
Shushma?
See, I'm always intrigued when mathematics is applied to biology. It does a lot of wonderful things. But the conceptual thing is that you like to generalize everything. And in biology, we think that every species and even within the species, every individual has some very specific traits and characteristics. And so flattening everything will also eventually lead to loss of the special characters. And that will have a much broader, like conceptual problem.
Yeah, I think it's
More like philosophical question at this point, but
yeah, I think it's probably important to think about encoding the species concept context. So when the model sees a sequence, it doesn't just think of it as a random DNA sequence, but it thinks of this is a DNA sequence from Arabidopsis, or this is a DNA sequence from maize, and perhaps even understands how closely related those species are. Because I think that's the key to understanding that perhaps, some X amount of generalization is possible across, say, the Anthropogony, but once you go from monocots to dicots, these things that I've learned to generalize more locally, I can't generalize, more distantly. So yeah, that's something we're working on at the moment in terms of understanding the species context of the sequences and their level of relatedness, which hopefully might go some way to addressing that.
I mean, we know a lot about the codon usage preference, right? In terms of the coding regions, and even if you have the similar sequences in different species, it's not going to lead to the similar expression.
Indeed, yeah.
Right. So that part is no, but what we don't know, how the promoter sequences and cis elements and other non-coding regions, what that variability eventually means.
Yeah.
So we have a little, I think, less insight, and perhaps the more data that comes from the epigenomics or that kind of the DNA folding models and things like that, perhaps will shed the more light on that. But yet we don't understand that.
Yeah, I think if we have models that can make those species specific predictions of, for example, gene expression, then that would allow us to take these kind of in silico approaches, where I showed that sort of shuffling of the nucleotides to see what changes it makes to the predictions. So I think if we were able to make species specific gene expression predictions, then those methods of adjusting sequences and seeing how it changes the predictions might start to give a way of providing some insight, to complement other experimental methods.
Yeah, and I think your insights and your predictions will be very good at, I think, when we correlate and think in terms of the pangenomes. So I think that will have a kind of a parallel, right? Something that is coming from the predictive models and what we know from the genetic diversity. So that will be very interesting. So thank you very much. Excellent talk.
Thank you.
David, there's a comment in the chat too. Not sure if you want to address that one.
Let me just see if I can. Is this the final one? Let's see. Slides.
Yeah,
Yes.
actually,
Sorry, but yeah. Let's see.
I was just kind of thinking about Shushma's comment, and I'm not 100% sure I understood it, but having worked with deterministic models of pathway biochemistry and those types of things, which compared to this are tiny. When we look at the genome, and I think it was the comment of losing the individuality. I don't think that you lose individuality. I think you find commonality. And you know, when you take it from really a neophyte's point of view, when you ask questions about comparing genomes, that there's a tremendous amount of similarity, obviously, among species, even widely varying species. And we know why. I mean, a lot of the proteins and enzymes and structures are the same from species to species. So I guess it's not really a question that much, but, you know, at what point will we have the, I guess for those of you that do this daily, do we have the computing power at this time to actually compare a large number of total genome sequences to each other, or are we still basically doing it by these smaller segments?
Yeah, I mean, just to share some insights from us, I guess, or from our point of view, we are limited to looking at those smaller genome segments, and I think that is a limitation, and obviously a limitation that we'd like to address, because a small sequence of DNA from outside of the whole genome context is obviously very limited. And also, I guess, just in terms of the question you put in the chat in terms of the similarities, I think those similarities are in some ways a difficulty for us in terms of understanding how to model them, how to model them accurately, because this work that I've described, or this approach that I've described so far, involves sort of putting individual different sequences into the model and training using those. But I think these massive overlaps are perhaps more of a difficulty than they might first appear, because if you have one sequence from maize and then you have another sequence from rice, and if they're 95% identical, you know, feeding them both into the model at the same time, it's hard for it to know what to do with it then. Or if we're doing this masked language prediction task I talked about, where we say what's missing, well, it can just look at, you know, you give it the maize sequence and it can just look at what it got in the rice sequence without really learning much. So in fact, dealing with the similarity and the fact that some regions of the DNA are very different and some regions are very similar, and working out how that impacts how we train these models, I think is something to look at.
Right. I think I understood that. Thanks.
Are there any more questions for David? Well, if no, I think we can thank David for presenting and accepting our invitation for the great talk. So thank you so much, all of you, for joining us. And yeah, as usual, I will be posting the video on our YouTube channel. Thank you.